Hello everyone, and welcome to today's webinar. I'm Taylor Hood, Digital Content Producer and Taxonomy Specialist for CNN, and I will be moderating today's event. This webinar is titled Absolute Molar Mass Analysis of Medical Device and Pharmaceutical Polymers, and is being sponsored by Wyatt Technology. I will now hand over to Gavin Smith.
Welcome all, and thank you for joining us. Today we would like to present several case studies regarding the absolute molar mass analysis of a few modern medical polymers, with a special look at bioabsorbables and the role that multi-angle light scattering has to play. The following case studies are actual analyses from the size exclusion chromatography lab of SGS Polymer Solutions Incorporated. Polymer Solutions was formed in 1987 by Dr. James Rancourt and has been solving difficult problems and finding solutions for clients in the defense, pharmaceutical, medical device, packaging, consumer product, and litigation markets ever since. In order to support the needs of our clients across many regulated industries, Polymer Solutions has developed a robust quality system, including ISO IEC 17025 accreditation, FDA registration, CGMP compliance, and DEA licensing. In 2015, Polymer Solutions moved to a newly constructed laboratory facility, custom designed for the specific demands of quantitative and qualitative analysis, as well as hosting expert witnesses and client consultation. Polymer Solutions Incorporated was acquired by SGS in June of 2018 in order to expand their capacity for great science and gain the expertise in polymer analysis offered by Polymer Solutions. Under the leadership of SGS North America, Polymer Solutions is continuing to deliver great science through a customer-focused approach, providing custom and innovative solutions to complex problems and an attitude of continuous improvement. In the size exclusion chromatography lab of SGS PSI, we are commonly asked to analyze the molar mass distribution of polymers in the food packaging, consumer products, medical devices and drug delivery systems, raw materials, biopolymers, and biological explant samples. Depending on the specific needs of our clients, we have options about how to perform molar mass distribution analysis. The simplest method for calculating the molar mass distribution of a sample is conventional column calibration analysis with refractive index detection, where well-characterized molar mass standards are used to create a calibration curve of log molar mass versus retention volume. This method may be adequate for a comparison between samples, but it is not a direct measure of molar mass. The results will be accurate only for the polymer used for calibration, and even the accuracy for said polymer is dependent on the accuracy of the molar mass value from the supplier's certificate of analysis. Additionally, the availability of column calibration standards is limited with regards to molar mass range and polymer type. By adding a multi-angle light scattering detector to a system, configured as shown here, you are able to add to the concentration source signal a light scattering signal and directly measure the absolute or true molar mass of the molecules eluting at each point of the chromatogram. Because all macromolecules scatter light by what is known as Rayleigh scattering, the molar mass of particles can be calculated based on the intensity of light scattered and the concentration of said particles. The benefit of this method is that you can accurately calculate the actual molar mass of your sample, even if standards are not available. Multi-angle light scattering analysis also allows for the direct characterization of large molecules, nanoparticles, and proteins by considering the differences in the intensity of light scattered at different angles. Certain considerations must be addressed for the proper performance of multi-angle light scattering analysis. Absolute molar mass calculations by light scattering require an accurate concentration source, such as UV-Vis or refractive index detection. The characteristics of each sample must be considered before beginning experimentation. If the sample is UV active, then the molar absorptivity must be known or measured in order to use UV-Vis as the concentration source. And a mobile phase must be chosen that will not contribute to light absorption at the chosen wavelength. For the use of refractive index detection as the concentration source, the differential refractive index increment of the sample must be known or measured. The differential refractive index increment, DNDC, is the change in refractive index of the solution when the solute concentration is changed. Good DNDC values directly applicable to your sample are often difficult to find in the literature because DNDC values are specific to the sample material, mobile phase composition, temperature, detector laser wavelength, and even the molar mass of polymer samples if the molecular end groups affect the refraction of light differently than the repeat units. It is often preferable for DNDC values to be measured under the exact conditions under which the analysis will be performed. The DNDC may be calculated from refractive index peak area, assuming 100% mass recovery, if the concentration is accurately known, no evidence of column adsorption or coelution interference is present, and the signal is strong enough to overcome any noise or wander in the refractive index baseline. Alternatively, if complete dissolutions of pure dry sample can be prepared at various concentrations, they can be infused directly into the flow cell of the refractive index detector, and the refractive index measured relative to pure solvent. This differential refractive index is then plotted versus analyte concentration, and the slope of a line fitted along these points is the DNDC. DNDC measurement can be particularly challenging when the sample is impure or contains an unknown amount of insoluble material. Despite these challenges, SEC-MOLS analysis remains the preferred method for characterizing the molar mass of large molecules. In our overview of the molar mass analysis of medical polymers, we're going to look at techniques and methods for analyzing therapeutic polypeptides used in pharmaceutical preparations, hyaluronic acid used in therapeutic injection formulations, and bioabsorbable polyesters used in leave-in medical applications. Our first case study sample is formed from amino acids naturally occurring in the body. Polypeptides are chains of amino acids, and in biological systems are the precursors to proteins. Therapeutic polypeptides have been introduced to the marketplace with some very promising results. Polypeptide drugs can be engineered to interact with certain biological receptors for very targeted responses. Because polypeptides are naturally occurring in the body, degradation byproducts are of little concern. Therapeutic polypeptides are currently being used in an array of medications, such as those designed for osteoporosis, chronic inflammatory diseases, obesity, and oncology. To begin our overview of the analysis of therapeutic polypeptides, we will cover some basics of SEC-MOLS experiments. Every SEC-MOLS experiment requires the same basic steps. Samples are dissolved and injected, molecules are separated based on size in SEC columns, detected, and the detector responses are used to calculate molar mass values. Every step must be considered carefully, and with polypeptides, the first two steps can be tricky. While most polypeptides can be solubilized in aqueous buffer systems, hydrophobic polypeptides may require the use of organic solvents or organic aqueous mixtures. The tendency of polypeptides to self-interact and form tertiary structures must be inhibited to prevent molecular aggregation. Additionally, the charged nature of most polypeptides can cause sample-column interactions, preventing the separation of molecules based solely on size. The polypeptide analysis we're discussing today is a confidential and proprietary hydrophobic polypeptide. In order to achieve total dissolution, prevent column interactions, and limit polypeptide intramolecular interactions, a specialty mobile phase of hexafluoroisopropanol, HFIP, containing 0.1% potassium trifluoroacetate was used for dissolution in SEC. HFIP is a highly polar corrosive solvent used most frequently for analysis of polar organic compounds such as polyamides or polyesters. Polymer Solutions has an SEC system dedicated to analysis in HFIP, which is enclosed in a specially constructed fume chamber for safety. In addition to the safety challenges of HFIP, this solvent is very dense. This must be kept in mind when planning the sample preparation, as all samples prepared in HFIP must be filtered rather than centrifuged. Following overnight dissolution, the polypeptide was fully dissolved and was filtered through 0.45 micrometer pore size PTFE syringe filters. As these samples did not contain insoluble filler material or express any evidence of insoluble gels, filtration through smaller pore sizes or through filters with glass microfiber pre-filters was not necessary. Additionally, the expected molar mass of the polypeptide was in the tens of thousands, low enough to be unconcerned about the threat of shear degradation from gentle filtration. The filtered samples were injected on PL-HFIP gel full-size SEC columns under standard SEC method conditions for data collection. For multi-angle light scattering, it is necessary, in addition to sample injections, to run a narrow molar mass distribution standard to align the detector signals by correcting for interdetector delay. Wyatt's Astra software also has a feature allowing for the correction of the sample band broadening as it travels between detectors. As you can see, the red-colored light scattering signal and blue refractive index signals are here misaligned, and the refractive index peak is very slightly broader than the light scattering peak. Even with minimizing the tubing volume between the two detectors, the sample band inevitably broadens as it travels from one to the next. By selecting this peak as the reference for alignment and band broadening, the software snaps the peaks into a near-perfect overlay. It is important that the standard used for this correction be narrow in molar mass distribution, so that the peak shape and apex retention times should truly align. We used a narrow 17 kilodalton polymethylmethacrylate standard for this correction. It is also necessary when performing multi-angle light scattering analysis to normalize the light scattering detector response. For this correction, an isotropic scatterer is injected, which is a polymer small enough in solution to scatter light equally in all directions, less than 10 nanometers in hydrodynamic radius. If, for a given peak, light is known to scatter equally in all directions, then detector voltages should be identical at all angles. For this, we use the same 17 kilodalton PMMA used for alignment and band broadening corrections. Once baselines and integration limits are set for the standard peak, the software is commanded to normalize the Mohs detector angles. And as you can see in this example, each detector voltage is multiplied by a normalization coefficient to match the voltage coming from the 90-degree detector. While some labs who perform only a single analysis repetitively on their SEC system may find it suitable to normalize their Mohs detector signals only occasionally, we are changing SEC columns, solvents, and sample types constantly, and so we perform band broadening, alignment, and normalization corrections with every analysis. These correction factors are applied to the configuration of all sample data files. These corrections are easily achieved with the single well-selected polymer standard. This standard is also well suited for system suitability evaluation. Run at the beginning and the end of the analysis batch, retention time and peak area reproducibility can be evaluated, as well as the accuracy of absolute molar mass calculations when compared with the supplier's certificate of analysis. With system corrections out of the way, processing of the polypeptide is a relatively simple matter of setting appropriate baselines and integration limits. The uniform peak shape with relatively little tailing seen here is indicative of a separation based solely on the size of the injected molecules and that extraneous interactions are not having a significant effect. The peak is well separated from any interference. The integration limits here are set to include the range in which there is adequate peak area from both detectors to calculate molar mass results. The Astra software then relates the light scattering data from all detector angles and the concentration data provided by the refractive index signal to calculate molar mass values. The differential refractive index increment used here to relate the RI signal to concentration was determined by infusing various concentrations of the polymer sample into the refractive index detector and measuring the response as described previously. As you can see in the presented figures, calculated molar mass values become more erratic and less realistic at the front and tail of the sample peaks. This is due to inadequate signal as the peaks begin and trail off. The effect of this erratic data is minimized by the low calculated concentration of these molar masses, but integration should still be limited to not include excessive amounts of irrational data. This is why integration on the right-hand side of the peak, as shown, does not include the entirety of the refractive index peak tail. This dataset indicates that the polypeptide samples being tested were particularly narrow in molar mass distribution. This is notable due to the effect this will have on the accuracy of calculated number and z-average molar masses. The M sub W, or weight average molar mass, is directly measured by light scattering analysis and is independent of the effectiveness of the chromatographic separation. The number average and Z-average molar mass values are calculated based on molar mass and concentration values for each slice across the chromatographic peak. Therefore, the accuracy of M sub n and M sub w are strongly dependent on the separation. For this particular set of samples, results were also calculated using conventional column calibration. As you can see, a significant difference exists between results calculated by moles and by column calibration. Any error in molar mass values calculated by moles associated with error in the D&DC value is dwarfed in comparison with the estimation of molar mass using standards. The molar mass values calculated from column calibration, if presented alone, would give a false idea of the character of the sample. Column calibration and light scattering data collected for these polypeptide samples also provide an opportunity to discuss the precision of the two methods. As you can see from the calculated molar mass values of 8 replicate injections of the 2 presented samples, results precision tends to be better with column calibration, which is useful in strictly comparative studies. However, even for comparative studies, moles may be preferable if the study is to be conducted over a lengthy period of time, as the elution pattern of a column may change as the column ages, and if different sets of columns are used over the course of the study, there could be differences in elution patterns that cause a shift in the data. Moles analysis directly measures M sub w, and therefore minimizes these effects. In review, all SEC moles experiments have the same basic steps of dissolution, injection, separation, detection, and results calculation. Detector signals must be aligned and corrected for band broadening using a narrow standard, and light scattering detector angle voltages must be normalized using an isotropic scatterer. Polypeptides can pose particular challenges for solubility and preventing column interactions. Be sure to check chromatographic data for evidence of column interactions in the form of severe peak tailing. Moles analysis is typically preferred over column calibration due to long-term reproducibility and the accuracy of molar mass results, as well as the lack of relevant standards for samples such as polypeptides. Our next medical polymer is quite a challenge to characterize for molar mass, though for different reasons than our polypeptide. Hyaluronic acid is a high molar mass mucopolysaccharide that exhibits nearly unlimited solubility in water, but is highly viscous in solution. Hyaluronic acid is commonly used in topical therapy, injectable formulations, wound treatment, and biolubricants for joints and eyes. Hyaluronic acid is used not only as a carrier in formulations, but also as an active ingredient. With its high molar mass and viscosity, certain considerations must be made for moles analysis. First, because of the large molecule size and weak chain bonding strength, hyaluronic acid samples are susceptible to shear degradation, not only during sample preparation, but also during analysis. Though sample filtration is a normal part of moles analysis in order to prevent small particles from creating noisy light scattering responses, it is not recommended that polymers over 1 million grams per mole be filtered. Low-speed centrifugation for 30 minutes will typically remove insoluble particles to an adequate degree. Tubing with an internal diameter of less than 0.01 inches should not be used for SEC systems characterizing high molar mass species. SEC columns must be chosen which will allow the polymer molecules to separate adequately, but do not put molecules under great levels of shear stress. Columns with large packing particle sizes are recommended not only to reduce shear, but also to reduce anchoring effects. Anchoring refers to polymer chains eluding later than expected due to their great size and their difficulty passing freely through the column packing, as shown by the theoretical red polymer molecule here. As polymer molecules pass through the beads of the column packing, very large chains tend to anchor. This may lead not only to extreme tailing and a lack of separation based solely on size, but also shear degradation for the polymer. This is exemplified in strongly tailing SEC peaks, such as the one shown. Molar mass plots may curve up significantly in the tail of the peak when anchoring occurs. Again, using columns with large particle diameters and pore sizes helps to reduce anchoring. Intramolecular interactions occur in hyaluronic acid samples when ionized carboxylic acid groups repel one another and cause the chains to adopt an extended conformation. The resulting high viscosity causes misshapen chromatographic peaks, an effect known as viscous fingering, something that is well known for hyaluronic acid solutions. Higher salt concentrations must be used to shield these electrostatic repulsions between sample molecules. The hyaluronic acid data presented here illustrates a marked improvement of peak shape and reproducibility when the sodium chloride content of the phosphate buffer mobile phase is increased. Higher salt concentrations alone are often not enough to adequately reduce viscosity. The combination of dilute sample concentration and higher salt content together reduce viscosity enough to obtain good data, while not diluting the samples to the extent that the concentration source is unusable. Injection volumes may need to be increased to accommodate low sample concentrations used to combat solution viscosity. You can see in the presented chromatograms injections of the same sample at 0.6 and 0.3 milligrams per milliliter on the same column using the same mobile phase. The sodium hyaluronate samples submitted by the client were dissolved in phosphate buffered saline and were analyzed on the SEC-MALS system at a slow flow rate of 0.5 milliliters per minute to reduce shear forces. A separate refractive index determination experiment was performed as previously described. Band broadening, alignment, and normalization corrections were performed using a 30 kilodalton narrow polyethylene oxide standard. These corrections were applied to all samples, and the baselines and integration limits were set for each sample chromatogram. An additional feature to consider when working with high molar mass samples is the Zimplot fit degree. The default for this feature in Astra is a first-order fit. The angular dependence of the Zimplot may become nonlinear, however, for very high molar mass samples. When analyzing samples expected to be over 1 million grams per mole, be sure to evaluate the validity of the default setting across the peak. It may be necessary to utilize a second-order fit. Shown here are the molar mass plots for duplicate injections of the submitted hyaluronic acid samples. Seen in the tail of the peak is the characteristically erratic results in the integrated region for which there is little instrument response from one detector or the other. In this case, however, the increase in calculated molar masses in the peak tail may not be attributed to low signal and detector noise alone, but may also be indicative of anchoring. The slight irreproducibility in the peak apex of duplicate injections shown for 3 of the samples is a good example of viscous fingering as discussed before. Despite these differences, calculated molar masses are consistent for duplicate injections where signal is adequate. The viscous fingering effect here is slight, indicating that a minor decrease in polymer concentration and/or increase in mobile phase salt content may optimize this method. Part of the challenge in method optimization is that further diluting the sample decreases refractive index response, and the results are subject to wonder, drift, and noise in the baseline of the RI detector to a greater degree. For example, a hyaluronic acid sample which poses much more difficulty may produce a chromatogram more like this, which is a sample from a separate analysis where the hyaluronic acid was supplied in an undisclosed dilute buffer solution, unknown to us in both composition and concentration. The large refractive index peak seen at the end of the chromatogram, which is extended to 80 minutes to minimize its effect, and also seen at the beginning of the chromatogram tailing off from the previous injection, is due to the apparently extreme difference between the mobile phase composition and the injection solvent, which was the diluent the sample was supplied in. This, combined with the low refractive index response due to low concentration, and tailing light scattering response possibly due to an improper salt composition in the injection solvent, serve as an example of the lower quality of data that may be created due to the challenges posed by hyaluronic acid samples. When given adequate information, however, very real and useful data is achieved. The results generated by these experiments, which you may notice produce rather broad chromatographic peaks, show that at roughly 1 million grams per mole, these samples were on the low end of what would be expected from hyaluronic acid. Mass recoveries were reasonably close to 100%, considering that there may be differences in moisture content, salt content, and purity between the samples. It is interesting to note that despite the broadness of the chromatographic peaks, the calculated polydispersity is rather low. This may be due to the inefficiency with which column packings of large particle size separate molar masses. This cannot be avoided, however, due to the size and susceptibility of HA molecules to shear degradation, as previously mentioned. This inefficiency in separation will have a negative impact on the accuracy of M sub n and M sub z calculations. The upward curvature in the molar mass plot in the tail of the peak, whether due to error in calculation from low instrument signal or from anchoring, also has an effect on the calculated number average molar mass, and so the calculated polydispersity must be taken with a grain of salt and is likely somewhat higher than is reported. To recap what we've learned from this hyaluronic acid analysis, it is important to keep in mind the expected molar mass of the samples to be analyzed by SEC. Shear degradation caused by preparation or analysis can be difficult to detect without method validation. Samples susceptible to shear should be centrifuged rather than filtered, and SEC columns with larger particle sizes must be used. Sample concentration must be considered in order to reduce viscosity, and the ZIM model fit should be evaluated for high molar mass samples. For charged polymers, mobile phases must be salted adequately to prevent intramolecular interactions, such as those that lead to higher viscosity in hyaluronic acid solutions. In our final case study, we look at the molar mass analysis of bioabsorbable polyesters. These polymers find themselves used in absorbable sutures, leave-in devices, timed drug release devices, drug delivery nanoparticles, and many more. Because of their involvement in medical procedures, these types of samples also become evidence in litigation when something does not behave as expected. There are many things to consider before beginning an analysis of a bioabsorbable polyester. Solubility is the first hurdle to overcome. For polylactic acid in particular, this can be challenging if you do not have the necessary information about the sample. Lactic acid exists in D and L configurations. The ratios at which these two are present in a sample containing PLA, and their arrangement in the molecule, have a drastic effect on solubility. Depending on the exact composition, PLA and polylactic coglycolic acid, that is, PLGA, samples may dissolve in tetrahydrofuran, a common SEC solvent that is easy to work with, or may require a stronger solvent such as chloroform or hexafluoroisopropanol. Another thing to consider that is affected by monomer composition grouping as well as the manufacturing process is sample crystallinity. Crystalline samples may require heat to dissolve, but most bioabsorbable polyesters are also at risk of hydrolysis, and so caution must be exercised. If significant heating is to be used, a heating time degradation study is recommended. Some polyesters may have acid or ester end groups, and acid end groups can cause significant column interactions if preventative measures are not taken. Copolymers like PLGA pose another challenge, and that is the consistency of the comonomer ratios across the molar mass range. If inconsistent, then the differential refractive index increment may be different at various points across the polymer peak. This would contribute to error in molar mass calculations and must be considered from the start of an analysis. As an example bioabsorbable polyester analysis, we'll look at polylactic acid, polylactic coglycolic acid copolymer resin. The PLA configuration present in these samples allowed adequate dissolution in tetrahydrofuran. Because we had information from the client expecting the samples to be of relatively low molar mass, the samples were prepared at 5 milligrams per milliliter, a higher concentration than we would typically use for SEC mols. The higher concentration is necessary for low molar mass samples because the light scattering intensity is related to both molar mass and concentration. In order to obtain adequate light scattering signal for lower molar mass samples, you must increase concentration. Following dissolution, the samples were filtered and injected on the SEC system under typical conditions, with an SEC column selected for the expected molar mass range. One item of particular importance on this slide is the use of formic acid as the mobile phase modifier. This is to prevent column interactions that may result from acid end groups on the polymer. Let's look at a brief example of this effect illustrated. An earlier PLA analysis produced some disappointing results for samples with acid end groups of approximately the same molar mass as PLA samples with ester end groups, as shown in Figure 1. We performed some investigative analyses with and without formic acid as the mobile phase modifier, with drastic results, shown in Figure 2. If analyzing organic soluble acidic polymers, acidic mobile phase modification may be necessary to prevent the ionization of the polymer and the subsequent column interactions. The PLA-PLGA samples in question were analyzed as mentioned, and the standard corrections were performed using a 30 kilodalton narrow polystyrene standard. In addition to the analytical functions described thus far, we took advantage in this analysis of the results fitting feature of Wyatt's Astra software. The results fitting feature allows for the extrapolation of the smooth curve that the molar mass elution would be expected to follow based on the smooth and stable region of the molar mass plot. This feature can be used to extrapolate at the front of the peak, tail of the peak, or both. This allows for the use of the refractive index data, which indicates the presence of sample molecules where the light scattering signal is too low to calculate realistic values. This is particularly useful for low molar mass species. As you can see, 5% or more of the peak would have to be excluded from integration if only realistic molar mass results were to be used without results fitting. In this case, only material greater than 2,000 grams per mole could be analyzed without results fitting. The results fitting procedure was applied consistently across all experiment injections, as shown in this sampling of chromatograms and molar mass plots. Note that the bottom right figure actually contains 4 separate injections, duplicate injections of a sample preparation at the beginning of the batch, and duplicate injections of the same sample preparation at the end of the batch, as a measure of instrument and analytical consistency. The non-Gaussian peak shape here is not due to viscous fingering as evidenced by the reproducibility between replicate injections, but is representative of the polymer chain size distribution. The numerical results confirmed the information we had been given regarding the molar mass range of the samples. In a comparison of the results obtained with and without results fitting, the results fitting proves to reduce deviation between replicate injections and prevents the low signal in the front and tail of the peak from causing falsely high calculated molar mass results. The difference is as high as 20% in some cases, particularly for the number average. In summary, some special considerations must be made for the analysis of bioabsorbable polyesters, as some polyesters may contain repeat units of various configurations and are often copolymers of more than one type of monomer. This poses challenges for solubility and differential refractive index increment accuracy. Some polyester samples are crystalline and require heat to dissolve, but may also be subject to hydrolysis if heated excessively. Organic soluble acidic polymers have a propensity for column interactions which must be inhibited by the addition of acidic mobile phase modifiers. Finally, the results fitting feature of the Astra software allows for the inclusion of data where inadequate light scattering signal leads to unrealistic calculated molar mass values. Thank you for joining us today as we have discussed the analysis of some common medical and pharmaceutical polymers. As you have seen, multi-angle light scattering analysis is truly irreplaceable for the complete characterization necessary for polymers in these fields. Though the analysis of these polymers can be quite challenging, with the right information, tools, and techniques, we are able to provide thorough, accurate, and meaningful data to clients in these fields. If after today's presentation and Q&A sessions, you have questions or challenges regarding MAL's analysis of polymeric samples, feel free to contact us at SGS Polymer Solutions, and we would be happy to help.
Hi everyone, so let's get started on our questions. Our first one is, can SEC-MALS be used for separation, identification, and characterization of highly branched polysaccharides, or do they have to be linear biopolymers or homopolymers?
Thank you. SEC-MALS can definitely be used for separation, identification, and characterization of highly branched polysaccharides. This is actually an area where the use of multi-angle light scattering can have an advantage over analysis using conventional column calibration, because the light scattering will measure the molecular weight of the polymer correctly regardless of whether it's linear or branched, whereas with branched polymers, they're more compact in size, so they may have a later elution volume on the SEC columns, which if you were just using conventional column calibration without MALS, you may be misled into believing that that branch sample has a lower molecular weight than it actually does if you are calculating molecular weight by comparison with linear calibration standards.
Our next question is, are the small peaks in the polypeptide chromatogram evidence of aggregation?
If you're referring to the small peaks that are very early in the chromatogram that elute well before the polypeptide peak. Most likely those are some kind of small particulates that are in the sample injection or in the SEC system in general. Sometimes when you make an injection, there are particles that are lodged in the columns that can be shaken loose just by the pressure wave that's created by the injection valve rotating and momentarily disrupting the column flow. Aggregation would typically be seen as a light scattering peak that is closer to the main polymer peak and would be eluting as a peak shoulder, or in the extreme, a large peak that dominates or dwarfs the light scattering signal from the main polymer peak.
How do you prevent aggregation?
So prevention of aggregation, a lot of times that can be done with appropriate selection of mobile phase, solvent, or modifiers. It depends on the type of polymer sample and the reason why it's aggregating. You can have aggregation due to hydrophobic interactions, in which case you would want to use a solvent that was nonpolar enough to prevent those aggregations. Or if you're dealing with polypeptides or proteins, sometimes it's acid-base interactions causing aggregation, in which case you would adjust the mobile phase pH or add a salt modifier to provide screening or shielding of electrostatic interaction.
How do you measure RG of macromolecules below 10 nanometers in size?
If you want to measure radius of macromolecules below 10 nanometers in size, that's below the range that can be measured with multi-angle light scattering in most cases. So you could use dynamic light scattering, or you could measure hydrodynamic radius using online viscometry detection.
Well, thanks for that one. Is there an advantage to using moles versus dual angle light scattering for polymers smaller than 100K?
Yeah, I don't know if there's really an advantage in one technique versus the other. The thing I like about moles versus dual angle light scattering, most dual angle light scattering instruments, the way they are calibrated is by injecting a polymer standard of known molecular weight, and then you use that standard to establish a calibration constant for your dual angle light scattering detector, whereas with a multi-angle light scattering detector, the way that those are usually calibrated is by infusing toluene into the flow cell. The calibration of the moles detector is typically more accurate, and the resulting molecular weight determination tends to be more accurate, which is not to say that you can't get accurate results with a dual angle light scattering detector. But you're depending more on the accuracy of the molecular weight measurement of the standard that you're using to calibrate.
Our next question is, how pure does the sample need to be? If there is a small amount of small molecules present, would the measurement still work?
So purity comes into consideration mainly for the measurement of the specific refractive index increment, or dN over dC. For that measurement, any impurities that are present in the sample will result in an inaccuracy in the measurement of the dN over dC, because if you have an impurity, if you're doing the online dN over dC measurement, the impurities will contribute to the refractive index that's being measured for the solution. So what you're getting is not the refractive index increment that's due only to the polymer in solution, but you're also getting an effect from the impurities. And then you also may not know the concentration of polymer accurately, which will also affect the accuracy of the dN over dC measurement.
Our next question is, can this be done via fluorometric detection? It may be a more sensitive detection when the targeted biopolymer is a low abundance analyte.
So if you're working with a sample that's so low in concentration that you need fluorometric detection as opposed to regular UV absorbance detection, then you're probably at a concentration that is too low to get an adequate light scattering signal for molecular weight measurement. And the other problem with samples that fluoresce is the fluorescence would be a problem for the light scattering measurement because the signal that you're trying to measure in the light scattering detector from the polymer scattering light would likely be overwhelmed by the fluorescent signal from the polymer. So yes, you could use fluorescence detection for a conventional or column calibration GPC analysis of a biopolymer, but I doubt that it could be used for a light scattering molecular weight measurement.
Our next question is there a minimum DN/DC value that is useful to measure a polymer in analysis?
There may be a minimum practical DN over DC value because the smaller the DN over DC value is, then the lower the signal-to-noise ratios of both the light scattering and the refractive index detectors. And the light scattering detector signal is proportional to the square of the DN over DC. So you may be able, if you have a very high molar mass polymer, then you may still have a strong enough light scattering signal if you're dealing with, say, a DN over DC of, say, hypothetically 0.01 milliliters per gram. I'd say that might be about the lowest that you could deal with and then still be able to get a useful measurement of molar mass using light scattering.
Our next question is, would the use of a small amount of linear alcohol with salt help in better dissolution of materials like hyaluronic acid?
I don't think that the alcohol would necessarily help with hyaluronic acid. Hyaluronic acid, to my knowledge, we don't have issues with hydrophobic interactions, which is what adding an alcohol would help with. I think with hyaluronic acid, it's more you're dealing with a polyelectrolyte, and it's typically a very high molar mass polyelectrolyte. So the issues with that are inter- or intramolecular repulsion of negative charges along the polymer backbone, which results in an extended chain conformation, which in combination with the very high molar mass gives you a very high viscosity of the injected polymer solution. So I think the main thing is to operate at a pH where the carboxylic acid groups are ionized, and then also have some salt present to shield electrostatic interactions.
Another viewer asked, how do we do molar mass determination of polymer blends, for example, PCl, PLA?
So polymer blends are very challenging to analyze. If you're using light scattering, then the question is the DN over DC of each polymer in the blend the same? If the DN over DC is significantly different, then you have a challenge because if those, typically in a polymer blend, there's overlap in the molar mass distributions of the two polymers in the blend. Now, if one of the polymers in the blend absorbs ultraviolet light and the UV absorbance for one polymer is significantly greater than the other at a given wavelength that we're able to work with, then we can use the light scattering, refractive index, and UV absorbance detectors together to deal with the polymer blend. The way that works is you use the UV absorbance detector to determine the concentration of one of the polymers in the blend selectively for each solution slice in the chromatogram, and then from that you can back out the concentration of the other polymer in the blend and then be able to figure out how much of the light scattering response is due to one polymer versus the other. And the Wyatt Astra software has what's called a protein conjugate analysis calculation. And that can also be used for polymer blends, but again, it depends on one of the polymers in the blend having a significantly greater UV absorbance than the other.
One viewer asked if you have any recommendation on the pore size for sample filtrations, as molecular weight may be a wide range from thousands to millions.
I think with sample filtration, you always have to be careful if you're dealing with high molar mass samples, and typically we consider something around 500,000 to 1 million grams per mole, where you have to start worrying about potential shear degradation of filtration. So in that case, we might go with a 0.45 micron pore size filter as opposed to a 0.2 micron. The other consideration is by increasing the diameter of the syringe filter, then you can reduce the shearing that the sample may be exposed to. So a 25 millimeter or 33 millimeter diameter syringe filter works better than a 13 millimeter diameter syringe filter for high molar mass samples where shear degradation is a concern. And then the other thing to do is just push the sample through the filter very gently and very slowly.
Another question that we've received is, what is the lowest molecular weight detectable by light scattering?
So if you will go on Wyatt's website, they have a large selection of application notes. And there are a couple of application notes on there dealing with analysis of polymers that are in the 500 grams per mole, 1,000 grams per mole range. The key to that is, number one, is that you choose a mobile phase solvent that maximizes the dN over dC value. And then the second thing that you can do to help is bump up the sample concentration. You can inject the sample at 5 milligrams per milliliter or even 10 milligrams per milliliter. You wouldn't want to inject such a high concentration for higher molar mass samples because you may end up with a sample solution that's too viscous and it won't distribute onto the SEC columns uniformly. But for low molar mass samples that are low in viscosity, then you can go to quite high a sample concentration. And having both a high dN over dC value and then also a higher sample concentration, both of those things increase the signal-to-noise ratio for the light scattering measurement and can get you into a range where you're getting a strong enough signal.
Why is there such a large difference in molar mass values between moles and calibration for the polypeptide samples?
So the reason for a large molar mass difference between light scattering and column calibration is that size exclusion chromatography or gel permeation chromatography separates molecules based on their size in solution and not necessarily based on their molar mass. So if you're using conventional column calibration with standards, then the retention time of your calibration standards is dependent on their size in solution. The retention time of your unknown polymer sample is also dependent on its size in solution. And the relationship between size and molar mass varies for different polymers based on their configuration, whether they are branched or linear, then also based on their conformation in solution. And conformation is really a function of the bond stiffness or the freedom of rotation about bonds in the polymer background or in the polymer background bone. So if you have for example, a good example is polyethylene, linear polyethylene, which is just CH2 groups that are are linked in series, there's nothing hanging off of that chain, and it's also fairly free to rotate versus polystyrene, which polystyrene has a benzene ring hanging off of each repeat unit. So a polystyrene chain is molecularly more dense. It has more mass per given chain length than polyethylene, so the two will have a very different relationship between molar mass and size in solution.
Another question that was asked is, instead of HFIP, you might be able to solubilize hydrophobic polypeptides with detergent in an aqueous solvent. What are the relative advantages and disadvantages of these methods, and how do they affect the SEC mols analysis?
So the mechanism by which detergents in aqueous solutions solubilize peptides is by the detergent forming micelles in solution. So the micelles have a hydrophobic pore, so they create a hydrophobic environment within the aqueous solution. So what will happen is the peptides will be solubilized inside of one or more micelles in solution. So if you attempt to perform a light scattering measurement of that solution, then you'll be seeing the light scattered primarily by the detergent micelles, and that will overwhelm the response from the peptide itself. So you won't be able to measure the light scattering response from the peptide alone, so you won't be able to measure the molecular weight of the peptide in the micelles. So HFIP works for SEC mols measurements of peptides because it is a solvent that can directly dissolve the peptides in solution. Now, there may be other organic solvents that will work for specific polypeptides. It just depends on the particular peptide that you're dealing with. So I think any polar organic solvents such as dimethyl sulfoxide, dimethylacetamide, dimethylformamide, NMP, those are all solvents that could work potentially for SEC mols of specific hydrophobic polypeptides. So my advice would just be to test solubility of your peptide in different polar organic solvents and then see what works, and then decide what you're going to try based on your own concerns for the relative hazards of each solvent.
We have one more question. Filtering versus centrifugation of polymers with molecular weight greater than 1 million. Is that a general rule for your lab or only applied to the hyaluronic acid work?
That's something that when we know that we're dealing with samples that are greater than 1 million molecular weight, and especially if we're dealing with water-soluble samples, that's where we definitely start to become concerned about shear degradation when filtering samples. Not to say that it can't be done successfully, it's just that's where we stop and think, well, should we centrifuge these samples instead of filtering them? Do we need to do some kind of a verification or a comparison of filtration versus centrifugation versus injecting the sample without filtering or centrifuging? And just we want to make sure that we're not degrading the sample due to shear degradation.
Well, that's all the time we have. Thank you again, Gavin and Jason, for your fascinating presentation.